So, we've learned a lot about what protein structure is, the hierarchy of primary, secondary, tertiary, and quaternary structure. We learned a lot about secondary structural elements like alpha helices and beta strands that interact to form beta sheets. And we've talked about fibrous proteins like collagen and keratin. But fundamentally, how do we know what protein structure is? How did they figure out 75 years ago that nature had it right millions of years ago to make the opposite handedness of fibrous proteins like collagen and keratin? Well, currently there are two main methods to determine protein structures. There's X-ray crystallography and then NMR spectroscopy. In lecture here, we're going to keep it fairly simple to understanding these two. And then in lab, we're going to understand some of the new techniques, particularly in Biochem 2 lab. We're going to understand how you can predict structure, how you can model structure, things like that with some new techniques. But fundamentally, X-ray crystallography and X-ray crystal structure determination has been around for a long time, not just for biological molecules, but what you need to have is you need to have a crystal of a molecule, right? It could take decades to crystallize a biological molecule, but basically it's a frozen image. And what you do is you bombard that crystal with X-rays, which essentially are electrons. And what happens is that those electron clouds around atoms are going to deflect or diffract the beams of electrons that are coming at it and essentially create a shadow of where atoms are. And then since you know the structure or the sequence, the primary sequence of your protein, you can then sort of map on, well, this structure, this shape sort of looks like it could be a leucine residue or a glutamic acid residue. But the limitations of this is it really requires a very well-ordered crystal structure, and it, again, could take decades. If you think about, like, if you make rock candy, for example, right? That's a question came up in class. Well, how do they make structures? Well, you have to have a solution generally of a protein, and then what you tend to do is try to evaporate the solvent, much like you do when you make rock candy, and you hope that these biological molecules sit very still and form these well-ordered crystals that can get good images from. It's very hard to do oftentimes for some proteins. NMR is a lot easier because it's a dynamic image. We'll talk about the pluses and minuses of that. But also all you really need to have is a soluble molecule in solution. And oftentimes that's fairly easy. You place, again, we learn about NMR, you place your molecules in a magnetic field, and atoms are going to feel interactions through space. So highlighting that this is a two-dimensional NMR technique that sort of has atoms feel where other atoms are. But the main limitation that you have is that you can't manage data for very large molecules. So you're sort of limited in size to either small proteins by themselves or looking at domains of proteins that maybe will fold independently such that you can study them as such. So again, some pros and cons of this. Sometimes people may argue that a dynamic image you get with an NMR structure is maybe more representative of reality, whereas when you force a molecule to sit still, is that really what that molecule looks like and behaves like in solution? Regardless of how a structure is determined, if you want to publish that structure, you need to provide the coordinates to what's called the protein data bank. We're going to look at the protein data bank in a minute. So solved structures are going to have coordinates that are deposited here. And why that's useful and why that's helpful is basically anybody who has a molecular visualization program can take the file essentially from the PDB and open it up in these visualization programs. Okay, so this is just another slide that sort of has this same information, right? X-ray crystallography, the advantages of it is you can have large molecules that you can investigate at generally very high resolution, particularly if those molecules sit still very well. Whatever the crystal or whatever the structure determination is, it's usually reported with a certain resolution. You'll see some number that has angstroms with it. Now, you can think of that as how much wiggle room do we have for where we think that atom is. So the smaller the number, the more confident you are in the position of that atom. So solved to a 0.1 angstrom resolution means I'm pretty confident that this atom is right here, plus or minus 0.1 angstroms. Again, the limitations that you have for X-ray crystallography, though, is that your molecule must be able to be crystallized, right? Which requires it to be ordered. You conformationally restrict a protein. Hopefully that rings some alarm bells. That's entropically unfavorable to make a large molecule sit still. But again, if you have a large molecule, that might be the only way to get a structure from it. Because one of the limitations, going to the other side here for NMR, is it's not useful for molecules that are larger than about 40 kilodaltons, which is about 120 amino acids, right? We're going to see this a lot. It's kind of equivalent to molecular weight, but for biological molecules like proteins. Okay, so one of the things you'll notice is that these techniques are sort of complementary, right? If you have a large molecule, it's not going to be able something that you can do sort of with NMR, but stop that for a second. So for example, if you have a molecule that you need to crystallize, but it's not crystallizing, an advantage of NMR is that you can look at that molecule in solution. It just needs to be soluble, right? So these are often complementary techniques in that regard. So a little bit more about NMR spectroscopy there. You'll see when you look at structures that are solved by X-ray crystallography, you usually see a single structure that's there because it's a molecule that sat still. I like to think and call NMR spectroscopy structures blurograms. I'm going to zoom in here so we can sort of see this. You'll see a bunch of structures that are overlaid. And what that allows us to see is maybe domains that have a little bit more dynamic nature, because as you captured these pictures of these dynamic molecules in solution, you might see that there's a lot more flexibility with a certain domain than another. So again, a huge advantage of NMR spectroscopy is that basically any molecule that can be in solution, you're able to see. And some might argue that sort of the real-time aspect of this might represent a more real structure of what we see. So proteins undergoing sort of dynamic motions, if you're thinking about protein folding dynamics and so forth. So X-ray and NMR crystallography are sort of two workhorses for determining protein structure. I'm going to go over here now to what we can see with our protein data bank. I'm going to talk about this in our next learning unit when we look specifically at structures of hemoglobin and myoglobin. But if you just were to type in PDB 101, for example, into Google, you could get to the Protein Data Bank's sort of training and outreach portal. This is a great resource if you think protein structure is pretty cool, you want to learn a little bit more about it. They always have a molecule of the month. So for October right now, this is pretty cool. We're going to learn about these next semester. These are the incretins, GLP-1 and G1P. So these are what we see with Ozempic and Zepbound and all of these sort of weight loss management drugs, right? They are agonists to these molecules. And so we're going to see these again actually next semester in Biochem 2. But you'll notice here you can see a lot of different things. You can see a little bit. So if we wanted to click on this stuff about incretins here, we can sort of see that these are molecules that are important for sort of maintaining blood sugar homeostasis. So again, they'll probably at some point talk about, here's these receptors that bind these. And so they probably will at some point sort of mention things like Ozempic and so forth. I haven't read through that yet. But we can see that there's some really interesting things. You want to learn a little bit more about structures and structure determination. You can sort of read some things here. Guide to understanding PDB data, right? You want to take a deep dive as to how structure is determined and looking at structures and so forth. We're going to do this a little bit more in lab and a lot more next semester, but this is a great resource to think about learning those different things.